friends, welcome to the Biological Life Sciences channel. Today we are going to see retroviral vectors and how these retroviruses are used in cloning experiments in genetic engineering. So let's begin. So let's see what are retroviruses first. These are the class of enveloped virus, means these viruses are having a coating. Since it is retro, it is also called as reverse transcribing virus. So there is a presence of reverse transcriptase enzyme here. It is having single-stranded RNA as the genetic material. Retroviruses are one of the mainstays of current gene therapy approaches. So whatever the gene therapy approaches are available now, these are on retrovirus-based therapies. The recombinant retroviruses such as mononemurin leukemia virus have ability to integrate into the host genome in a stable fashion. As we know, the infection by retroviruses is stably integrated into the host genome in many organisms, eukaryotic organisms. So therefore, these viruses are used as a genetic engineer, that is its ability to transfer the genes into the host cell. Let's see the whole cycle of this virus. We are taking an example of a HIV virus here. You can see the HIV virus here. It attaches to a particular receptor called as CD4 receptor along with the core receptor CXCR4, CCR5. Now these receptors are present on cell membrane of lymphocytic cell. Now using this receptor, this virus fuses with the cell membrane, that is HIV fusion with the host cell membrane occurs here, and the metric protein and genetic material is released into the cytoplasm. You can see here HIV core is disintegrated here and released into the cytoplasm. The HIV RNA is then taken into the cell and using a particular enzyme called as reverse transcriptase shown here in red color, it is converted into the double-stranded DNA. Now this double-stranded DNA is the cell recognizes this double-stranded DNA as its own material, and it is not able to differentiate into own DNA and the viral DNA, and this viral DNA enters inside the nucleus. This viral DNA integrates with our host genome. Now the host cell thinks that this genome is its own DNA and it starts transcribing this. As it transcribes it transcribes all the machinery required for the new virus formation, and this mRNA is formed. Now this mRNA comes out of the cytoplasm and it is again translated, and new viral proteins are formed from this, and these viral protein then transfers to the or migrates to the cell surface where it forms the viral assembly here, and you can see here how the virus is embedded, and this RNA copy travels in a protected manner to this membrane assembly and gets embedded in this, and thereby the virus is budded out from the cell. This is how the cell acts as a factory for the synthesis of these kind of retroviruses. So this is the whole life cycle or the infection of virus, and the mature HIV is formed here, and this virus is ready to infect another cell. So here the lymphocytic cell is acting as a cell factory for the synthesis of HIV virus. You can see here the infection cycle is again shown here in a cinematic view. First of all, there are few steps here. First step is adsorption to the cell membrane, then penetration. This virus penetrates into the cell, uncoats its genetic material. The biosynthesis takes place of the viral RNA into the DNA. This DNA integrates into the host cell and the biosynthesis starts. It is transcribed into mRNAs and viral proteins are formed. These proteins migrate to the cell surface, and this RNA then goes under that surface and it bulges out or the assembled and is assembled and released from the cell. So this is the infection cycle of HIV. Let's see the genome in details. Retroviral vectors, these are the commonly used and known to integrate the genome of infected cell in a stable and a permanent fashion. You can see in the infection cycle of HIV, the viral RNA is successfully integrated into the host cell, so that property of these retroviruses are used to integrate different desired gene into the host cell. Reverse transcriptase in virus allows the integration into the host genome. The enzyme is present itself in the genome of virus. We are going to see the genome in details. There are two types of retrovirus or retroviral vector, that is replication competent and replication defective. So we want replication defective vector, as we don't want that virus to replicate again, but we want it to transfer our gene of interest into that host cell. Usually the replication defective virus are preferred in practice, as they allow for several rounds of replication due to their coding regions only, and it does not code the virulent part of it. Let's see how it is constructed. That is defective retroviral vector. Retroviral vectors are derived from wild type retrovirus, that is mononemurin leukemia provirus, and are engineered to carry a foreign gene of interest into the target cell. The retroviral vector are composed of the genes of interest, the cis-acting elements of the viral genome, and the removal of trans-acting elements genes, that is GAG, POL and ENV. The GAG, POL and ENV genes are virulent genes here. Let's see the details of a genome. The viral genome is having LTRs at both ends. This LTR is long tandem repeat that helps in protecting these genes. There is a presence of 3 main genes here. The cap is present at both ends, and this also helps in integration. Now, the GAG, POL and ENV, these are the GAG gene responsible for different components of the protein of viruses that forms matrix. The polymerase is responsible for reverse transcriptase, integrase, permease enzyme, etc., which helps in synthesis of viral particle inside the cell, and the envelope gene is responsible for formation of envelope protein. Now, this part can be removed from these viruses. Let's see how. You can see here this virulent part from the retrovirus is removed, and our fragment of interest is added into this. Around 1 kb of genome can be added here maximum, and this is packed again into this virus. So this retrovirus is having property that it can be again packed in lab. Okay, so the assembly can be reassembled using this genetic material. Now this is having our fragment of interest, that is cloned gene. Now when this virus infects a particular cell, it will transfer our particular gene of interest into that infected cell. So generally these for example HIV, these kind of viruses infects the immune cells. So therefore the specific genes which are required in immunotherapies can be added into these viruses and transferred into the immune cells of our body, which helps in gene therapy. So this is how a gene is transferred into the retroviral, transferred using retroviral vector into the host cell. There is a lots of application, as I told you. In this you can see the gene of interest is added here, healthy gene is added, and the defective gene is removed, and now it is injected into the cell. This virus then delivers this healthy gene into the cell. Now again, as a part of its infection, it will transfer this gene to all the immunogenic cells into the body, and it will transfer the copy of healthy gene of a particular component, for example synthesis of erythropoietin or synthesis of certain antibody. It will transfer it to all immune cells and thereby correcting the defect. So it has vital applications in gene therapy and animal tissue culture. So this is how the retroviruses are used. There are different limitations also. Monthly retrovirus involves the requirement for cell to be actively dividing for transduction. So cell should be in a actively dividing state here. Insertional mutagenesis due to the integration into the host genome might lead to the cancer or leukemia. In certain cases, what may happen? It may lead to leukemia, as this viruses constantly mutates. So suppose it mutates at a wrong position in the infection cycle, it may lead to the cancer of leukemia. So therefore direct application into the human being is not allowed here. It is first of all tested on different cell lines, and then only it is approved for humans. Thank you. If you like the video, please press like button, share, comment and subscribe. Thanks for watching.